Hi everyone. We're going to cover our last chapter for this quarter, chapter 23, which is all about carbohydrate metabolism. So we're going to start by going through a general overview of what's called cellular respiration. So in cellular respiration, we have a set of metabolic reactions or processes that are converting chemical energy from, for instance, glucose or other molecules into something called ATP. So we'll talk about this molecule ATP later, but basically ATP is really important for energy storage. So then we could use that energy somewhere where it's needed later on. So let's just look at the different steps involved in cellular respiration. So the first step is glycolysis. So this is where glucose or even other monosaccharides will be broken down into two pyruvate molecules, or rather they're converted into two pyruvate molecules. Then once we have that pyruvate, that's going to go through pyruvate processing and become a molecule called acetyl-CoA. Then acetyl-CoA is going to enter something called the citric acid cycle. This has also been known previously as the Krebs cycle, but we've been moving away from naming things after scientists, especially if they're problematic, so now it's more often known as the citric acid cycle. And then finally, we have the electron transport chain and chemiosmosis. Okay, so again, the main purpose of cellular respiration is to break down carbohydrates into smaller molecules and specifically store energy into a molecule called ATP. So we're going to talk about all of these processes, but let's start with glycolysis. So when we consume carbohydrates, we immediately start to digest them. And the main reaction that's used to digest sugars or carbohydrates is hydrolysis. So we've seen an example of hydrolysis before, specifically with sucrose. Remember, sucrose is made up of the monosaccharides glucose and fructose. So if sucrose reacts with water through hydrolysis, it's actually also reacting with that enzyme sucrase that we've talked about previously. And eventually we get the monosaccharides glucose and fructose. Now, this can also occur with other carbohydrates. So if we have a polysaccharide, for instance, like starch, starch is made up of multiple glucose monosaccharides. So after digestion through hydrolysis, we're just going to end up with a lot of glucose monosaccharides. Lactose is found in sugar, and after digesting lactose, we end up with glucose and galactose monosaccharides. Maltose, after it's digested, becomes glucose. So once we've produced these monosaccharides, they can then go on to that first process, glycolysis. So carbohydrates account for 45 to 55% of daily energy needs in the typical American diet. So once carbohydrates are digested into glucose, fructose, and galactose, they're then absorbed into our bloodstream through the lining of the small intestine. Then they're transported to our liver. So in the liver, fructose and galactose are rapidly converted to glucose. So there's a common theme here. We're trying to get everything to become glucose so that we can then go through the process of glycolysis. Now, sometimes that's not possible to go from, for instance, a monosaccharide to glucose. But you could also have these carbohydrates converted into compounds that are metabolized by the same pathway as glucose. So as long as it's something that can enter the glycolysis pathway, then that's good. So blood glucose levels are really important to know and understand, especially in health-related careers. Blood glucose levels are also known as blood sugar levels, and this can be really important, for instance, if somebody has diabetes or if you are worried that they might have diabetes, you might measure their blood glucose levels and other things like ketone levels, etc. But blood glucose levels are measured after a fast of 8 to 12 hours because you want to see how the body is functioning without a constant supply of sugars. So blood glucose levels are highest about 1 hour after a carbohydrate-containing meal, and then it returns to normal after 2 to 2.5 hours. Now, these glucose levels are regulated by the liver, but we've also talked about previously how the protein insulin helps regulate glucose metabolism. So off to the right, you can see a chart for blood glucose concentration. And this is measured in milligrams for every 100 milliliters. So it's a small amount, but depending on how much is in your blood, that can make a huge difference. So after fasting for 8 to 12 hours, you should have a blood glucose concentration anywhere from 70 to 110 milligrams per 100 milliliters of blood. If you have anything below 70 milligrams, then you might have something called hypoglycemia, and this is where your blood sugar level is too low. If your blood glucose levels are above 110 milligrams, you might have hyperglycemia, which is a blood sugar level that's too high. And these are just ways that doctors can try to rule out different illnesses or diagnoses and kind of narrow down what might be going on if you're going to see them for a particular reason. All right, so glucose, we talked about previously. So glucose is what's called catabolically oxidized through a mini-step process to two molecules of something called pyruvate. So glucose has 6 carbons, and pyruvate only has 3 carbons, so it's almost like we're kind of splitting glucose in half, but it actually goes through multiple steps to become pyruvate. So we'll look at those steps in a second. Now, in addition to the 2 molecules of pyruvate, we also produce 2 molecules of that ATP we talked about, and we'll go over that in more detail. We also produce 2 molecules of NADH, 4 molecules of H+, and 2 molecules of water. So the net reaction for glycolysis is shown below. So the main thing to focus on here is that glucose eventually becomes 2 pyruvate molecules. Now let's talk a little bit about ATP and that NADH molecule, just so you can kind of get a feel for what their purpose is, because it is going to come up again. So ATP is also known as adenosine triphosphate, and this is the primary carrier of energy in our cells. So the structure for ATP is shown below, and notice that it's really similar to the structure for a nucleotide in RNA. So notice we've got the sugar ribose, and then we have a nucleotide base, and instead of 1 phosphate group, we actually have 3 phosphates that are all bound together. So this is where that triphosphate comes from in the name. ATP stores energy that can then be used later on, and the stored energy is primarily in the phosphate group. So what happens is a phosphate is removed through hydrolysis. So we just kind of chop off one of those phosphates, and that's going to release energy and produce something called ADP, which is adenosine diphosphate. So it's the same molecule we see above, just with one less phosphate group attached. Now, NAD plus or NADH is referred to as nicotinamide adenine dinucleotide. So NAD plus is a key component of energy production in the cell as well, and it's the principal carrier of electrons during cellular respiration. So remember we said that the last step in cellular respiration is the electron transport chain. So NAD plus can carry electrons all the way over to that last step, the electron transport chain, and we can then produce more energy. Now, the main difference here between NAD plus and NADH is the addition of a hydrogen to this carbon right at the top of the nucleotide base that we see there. So initially there's just one hydrogen there, but when we add another hydrogen, we're also going to add an electron as well. So that's where we can store those electrons for use later on. Okay, so let's look at the process of glycolysis, so glucose becoming 2-pyruvate molecules. Now, earlier we made it look really simple, right? But there's actually a lot of steps that go on in this process. So in the first step, glucose is converted into something called glucose 6-phosphate. And we actually use ATP to help provide energy for that process. So notice that ATP is going to get rid of one of its phosphate groups to glucose. So ATP loses a phosphate bonds to glucose, and then we have ADP at the end. In the next step, glucose 6-phosphate becomes fructose 6-phosphate, so it just changes slightly. And then after that, it becomes fructose 1,6-bisphosphate, which means that we have 2 phosphates. So bis means 2. And notice again, we're using ATP to provide some energy and another phosphate group. So it's going to become ADP again. Okay, and then after that, there's a couple of different pathways here because fructose 1,6-bisphosphate is going to break apart. So it's going to break apart into something called dihydroxyacetone phosphate and then glyceraldehyde 3-phosphate. But notice really quickly, dihydroxyacetone phosphate turns into glyceride 3-phosphate. So we end up with the same 2 molecules at the end there, but one of them had to go through an extra step to get there. Now, both of these glyceraldehyde 3-phosphates are going to become 1,3-bisphosphoglycerate. And what's really cool here is that NAD plus is going to come in and become NADH and help that process along. Okay, so now once we have 1,3-bisphosphoglycerate in both sides, we're going to use ADP to remove one of those 2 phosphate groups. So now ADP is going to take away a phosphate group and become ATP again. So that happens on both sides. Then we end up with 3-phosphoglycerate, which will become 2-phosphoglycerate. That becomes phosphenyl pyruvate. And then another ADP molecule comes in. It's going to take that phosphate group from phosphoenol pyruvate and become ATP. And then we end up with one of our pyruvate molecules on both sides for a total of 2 pyruvate molecules. Now, I'm not going to make you memorize this whole process, but I did want to walk you through it just to show you how complex it is. Our bodies are constantly doing this. It's taking glucose and going through all of these reactions to become pyruvate. So there's all these different steps involved, which is crazy to think about. Okay, so there are also other monosaccharides that can enter the glycolysis pathway. So fructose, for example, enters glycolysis as that dihydroxyacetone phosphate that we talked about, and glyceraldehyde 3-phosphate. So if we go back, fructose is going to enter here. All right, and then galactose enters glycolysis as glucose 6-phosphate. So let's go back. So galactose enters here. Now it's first converted into glucose 6-phosphate, so it doesn't enter as galactose. So it does go through a step first before it enters. So we are able to digest multiple different monosaccharides, but they have to be converted into something that's within the glycolysis process first. All right. Okay, so let's review. What is the main product of glycolysis? And why is carbohydrate metabolism important? What role does it play? So see if you can answer these questions on your own first. You can pause the video, and then when you're done, you can unpause and we'll go over it together. Okay, so what is the main product of glycolysis? What did we produce at the very end? Pyruvate. We also did produce molecules such as ATP and NADH, and those are going to go on to do other things as well. Now, why is carbohydrate metabolism important? What role does it play? And this can be different depending on how you interpreted the question. I would say that carbohydrate metabolism is important because it provides energy to our cells. So carbohydrate metabolism is important because it provides energy to our cells. All right. So now let's talk about the fate of pyruvate. Now earlier I said that it will become something called acetyl-CoA, which will then go on to the citric acid cycle. But there are other pathways that pyruvate can take. So we said that after glycolysis, pyruvate can be turned into acetyl-CoA, and it's actually oxidized into acetyl-CoA. So this would occur under what are called aerobic conditions. So this is where oxygen is present. Now this makes sense, right? Because if pyruvate is oxidized, that should mean that there's oxygen present. Not always, but typically. Now there's other ways that pyruvate can direct itself in our bodies. Pyruvate can also be reduced to something called lactate, and this would be under anaerobic conditions. So anaerobic means that oxygen is not present. So the pyruvate to lactate reversible reaction occurs typically when we are exercising very vigorously and our muscles are starting to get tired and they're producing lactic acid. Lactic acid can then be turned into pyruvate, and vice versa. That reaction can go back and forth. Now there's another process where pyruvate can be reduced to ethanol, and this is also an anaerobic condition. This is for some prokaryotic organisms, which we haven't talked about, and it's not super important here, so don't worry too much about it. But I just wanted to show you that pyruvate doesn't have just one path that it takes in our bodies. It can go on to other paths as well. Now, one thing to note is that all processes must regenerate NADH from NADH so that glycolysis can continue. Because we do need that NAD plus in glycolysis to keep the reactions going. So if we kind of break down the glucose to pyruvate reaction, remember that overall we were using 2 NAD plus molecules that sort of towards the end, and those were becoming 2 NADH molecules. Additionally, we used 2 ADP molecules, and this P here is just phosphate, and those became 2 ATP molecules. All right, so let's look at the different fates in a chart, because sometimes that's easier to visualize. So again, pyruvate has 3 different possible fates. Under aerobic conditions, which is what we're looking at, pyruvate can become acetyl-CoA, which will then go on to the citric acid cycle, and it actually is completely oxidized into carbon dioxide, which is what we breathe out. Additionally, pyruvate can be converted into lactate under anaerobic conditions, so without oxygen, or it can also be converted to ethanol under certain anaerobic or in certain anaerobic organisms. Okay, but we're mostly going to focus on this process, the aerobic conditions. All right, so next time we're going to talk about the citric acid cycle. So we said that pyruvate can be oxidized to acetyl-CoA under aerobic conditions, so conditions where oxygen is present. And then we'll see what happens after that. So I will see you in the next video.